Good evening, everyone. Once again, I welcome you all to this short-term training program under the NHP CAST. And today we are going to have our first talk under the theme Science for Future. And we are going to have this talk by one of the renowned scientists, Dr. Srinivasan sir. And he is a very fabulous orator and teacher for all of us. So with this brief note, I request my coordinator to please welcome Dr. Srinivasan sir and Dr. Archana Sachdev ma'am with a flower bud.
Oh, thank you very much. Thank you, Sneha.
So now I request Dr. Archana Sachdev ma'am to please introduce today's speaker.
Good evening, everyone. I feel very humbled to introduce the speaker of today. He's an excellent teacher. You will really enjoy his lecture today. And Dr. Srinivasan has been our teacher as well. So I can vouch for it. There can be nobody better than him when he takes a class. And Dr. Srinivasan is from Banaras Hindu University. He did his BSc, MSc, PhD from there. And he's a master of enzymes. He's got the wonderful basics which he can clarify to all of you in his classes. And Sir has been a principal scientist in the National Institute of Plant Biotechnology. And after that, he was a professor there. And after that, the project director, rather director of NIPB. And when he retired, he joined as an emeritus scientist. And soon after, Sir has joined us in our division as the chief editor for our journal, Journal of Plant Biochemistry and Biotechnology. And with Sir coming in this line, he's got a track record of being an excellent reader also. His research propensity is just too good. And Sir, whatever I say is too less for you. So I would say...
That itself is exaggeration, but anyway.
No, that's not an exaggeration. They'll all witness it now.
Okay.
So for past, present, future, and forever, enzymes, you will witness it today. What I really mean to say from my heart, that he's excellent. So let's start, and you see it yourself. See it for yourself, right? So please, welcome.
Thank you. Thank you, Archana, for your nice introduction. Most of it is exaggerated, partly because she has been our student and colleague. I would not like to keep this as a lecture, so I would not like to call it evening lecture. I would like to call it evening discussion, so please feel free. I am not bound by the timing of what I have been told, neither you should be. So I'm not going to tell you something extraordinary which you already did not know. But the unfortunate part is that something which is so common, which you see every day around you, you become kind of immune to it. You are not aware of the amazing properties and happenings which is all strewn around you. And that is what I'm going to talk to you. I took the liberty of modifying the title which Dr. Ranjan gave me. He said, the future for science, and he said, talk about future of enzymes. When I looked at the title, I realized that enzymes have been there since time immemorial. So I changed it that for past, present also, and in future. Enzymes, you will notice, will be present and have been exploited extensively. The way humanity is progressing towards, wherever it is, ultimately, it will all depend upon enzymes. It still depends on enzymes. I will illustrate this with some examples. So what I intend to discuss with you, I have just listed it here. I said the first point which is evident to all of you is absolute necessity of enzymes for any life process. First, please keep this in mind, we always tend to forget. The other is, I will talk to you about some of the amazing properties of enzymes. which are totally different from what you find in other chemical reactions or any other chemical agent or molecule which I have known of. It's amazing. I'll come to that. And then its application in you name a field, whether it is health, archaeology, agriculture, biotechnology, medicine. Enzymes have found applications, and they have been used since time immemorial. Knowingly or unknowingly, we have been dependent on enzyme for various day-to-day activities, and also will continue to depend on them. So I'll give you some examples. So these are the examples which I have selected are just indicative of the enormity of the application of enzymes and also the properties of enzyme, which are very amazing. Let me now go one by one. One, as I said, absolute necessity of life. So life, as you think today, cannot exist without enzyme. So if we take a bacteria or amoeba or a human being or now extinct dinosaurs, anything which flies, lives, multiplies, needs enzyme for conversion of some material into energy, synthesis of RNA, DNA, you name it, synthesis of the molecules which surround its, let us say, membrane formation. Every step requires enzymes. So if you think of that we will have life without enzyme, no, it's not possible. Okay. Now, so enzymes, what I'm going to talk about today are proteins. There are some enzymes, some other molecules like RNA which act as an enzyme, but that's a very limited one. Totally our discussion today is focused on enzymes which are proteins composed of amino acids. So it is something like 26 alphabets. In English alphabet, you have 26 letters. So is in the language of protein, you have just 20 letters. And all the life which you see around you, whether it is a rose plant or a bacteria or sequoia tree, eucalyptus, anything which you can think or see is all interplay of these 20-letter language which organisms speak. Okay? So you can produce a Shakespeare, you can write Bible, you can write a poem, you can write a short skit, you can write a comic page, anything is possible with those 20 letters. You can write a book on biochemistry, you can write a book on entomology, and so on and so forth. Just 26. Here we have 20 letters of the alphabet which play this enormous ability. And one of the properties of enzymes, one group of such proteins is what we call as an enzyme which catalyze reaction. So bear that in mind. Okay. Now, what are these amazing properties? One is high rate of reaction. You would dismiss this. What is this high rate of reaction? I'll give you some examples of some of the reactions which all cells catalyze, which are almost impossible. So let us say if you take that chemical, keep it in the bottle, what we call as a half-life, how much of half of that would be converted in molar terms is something, in some of these reactions, runs into billions of years. That means if you leave that chemical as such or as aqueous solution of that chemical, it will take the product which we see every day happening in all the bacteria, all the organisms, in all the cells. Look at the bacterial cells which has to multiply within 20 minutes, make it double. Everything has to be doubled. And that reactions are taking place which would have otherwise taken billions of years. Not one, not two, several of them. And that is the power of enzyme, that they have been accelerated, that they are happening in a fraction of a second. I'll give you some example. The other is high selectivity of enzyme. Selectivity, some of them are selective to the extent of high specific regio and stereospecificity of the substrates. Some enzymes are specific to the bond on which they act, which hydrolysis of which they catalyze, and so on and so forth. So in some cases, you will have enzymes which exhibit broad specificity for, let us say, when we talk of DNA polymerase, it can take up all the nucleotides, or an exonuclease which cleaves off DNA without any specific requirement, But if you talk of endonuclease restriction enzymes, they are highly specific, and so on and so forth. There are n number of such examples. And the other enormous property which is amazing is ability to carry out reactions at normal temperatures and pressure. If you want to carry out a chemical reaction, you generally see that chemists employs higher temperature, higher pressure, higher concentration of substrates, removes the product constantly. Here in a cell, if you look at the volume, there are thousands of reactions simultaneously going on in the same milieu. There are reactive groups all around, aldehyde, ketones, hydroxyl group, acid, alkali, everything together, happening together without confusion. That's another amazing property of enzyme, that they can get regulated. As and when the activity of a particular enzyme is required, it's active, otherwise it is shut off instantaneously. There are other mechanisms of synthesizing the enzyme proteins, which I'm not going into, about transcriptional regulation and so on and so forth. What I'm saying is ability of these enzymes to be regulated based on the need of the cell. They respond to number of the other metabolites. One metabolic pathway, for example, let us say carbohydrate metabolism, feeds into amino acid metabolism, amino acid feeds into fatty acids, and vice versa, and so on and so forth. So everything is interconnected. It's something like if somebody looks from the top at, let us say, at Delhi as a metropolis, he would be amazed. What is happening? There are so many people milling around, going, so many vehicles, so many, everything is happening, but everything is happening in a systematic way. From top, it will look random, unregulated. But in fact, at the micro level, they're all being regulated. Okay? So these are the points which I'm going to elaborate upon, and then come to some of the example. Is it okay? Any questions so far? None? Good enough. Let me talk about the rate enhancement. Look at this reaction, which is conversion of OMP to UMP, which every cell has to carry out, whether it is a bacterial cell or animal cell, plant cell. And the half-life of this is 78 million years. That means if you keep a solution of OMP, it will be converted to UMP 78 million years. In the cells, bacterial cells, it's converted in fraction of seconds. So that is the rate enhancement which enzymes have achieved. Another example. There are several examples. I'm just giving you two here. This is another one which is required for synthesis of, one of the steps which is required for the synthesis of hemoglobin in animals and chlorophyll in plants. Conversion of pyrrole 3-acetate to 3-methylpyrrole. This is a decarboxylase reaction. And look at the half-life, 2.3 billion years. So, amazing. And the enzyme has been, so look at what these enzymes had to overcome. This example really exemplifies what these proteins had to overcome to create life on this earth. A reaction which otherwise would not go, they have been able to convert it to such a way that it takes place in fraction of a minute. And it's happening in all your body, every animal's body, every plant's. So it's tuned all around us, each one of them. So these are just two examples, but almost all the enzymes, nothing is spontaneous except maybe dissolution of carbon dioxide into water. Again, you have carbonic anhydrase for it, which enhances it, and so on and so forth. So protein hydrolysis, if you just take proteases are so common, it would take at 25 degrees about 400 years. That's the half-life. Look at polysaccharide hydrolysis. So which has been converted into, even at 100 degrees, it will take 160 years. But these are happening in, as I said, fraction of seconds. So this is one of the important properties of enzyme, ability to enhance the reaction, enormous enhancement to overcome whatever these obstacles were to create life on this earth. Another, as I said, normal temperature and pressure. You are familiar with this process, Haber's process of nitrogen fixation, which is one of the major consumers of fossil fuel on this earth, because we generate a lot of fertilizer for production of ammonia. And look at the pressure. and temperature, 450 degrees and 600, and about 1000 atmospheric pressure is required with some catalyst for conversion of hydrogen and nitrogen into ammonia. Look at the bacteria, free living as well as symbiotic, which are strewn all over this earth, are able to fix nitrogen at room temperature without any extra temperature pressure. Don't you think it's amazing? This is one example. So all these reactions which are taking place at normal. If your body temperature is 37 or 36 point something, the reactions takes place. Those organisms which live in Arctic or plants which are at low temperature, high temperature, these reactions are taking place. Almost all the reactions, vital reactions. So enormous ability to function without requirement of a high temperature or pressure, which is required for an organic chemist. The other, which I am always fascinated by, is the ability to recognize differences, subtle differences in molecules. Here is, for example, you are already aware of number of D-glyceraldehyde, L-glyceraldehyde, D-amino acid, L-amino acid. Here is example of L-alanine and D-alanine. It's almost, I always used to wonder when I was a student that why is not, you don't get both these so-called enantiomers. If you talk to a chemist, if he carries out a reaction, he will get a mixture of these two. If the molecule exists as L and D, he will get a mixture which is called a racemic mixture in a reaction. But nature always prefers one and does not touch the other. In some cases, if it does, there are some cases where it does. There are some cases where the other enantiomer acts as an inhibitor. So nature, by design, by chance, has selected D-glucose as the substrate for life in most of these glycolytic pathways. So it is D-glucose, it is not L-glucose. Similarly, it has somehow selected the L-amino acids for the construction of proteins, and it stays as L-amino acid. Though the molecule, if I place the structure before you, I brought these to illustrate the point. What is called as a stick and ball model. Is this the same? No, this is the same. All the three identical. Can you see it here? Let us say that this is hydrogen, this is CH3, this is NH3, and this is COH. So this is alanine, alanine, alanine. Do you think the structure is same? All the three are same? Which one is different? This one? How do you know? How did you know? The way you do it is that you place this here and look at whether these are mirror images. Okay? So they will be, if you have a mirror image, okay, let us, they are not, okay, but one of them would be. So you have what are called as non-superimposable mirror images. This is a mirror image, and this is superimposable or non-superimposable. This is non-superimposable mirror image. So this is one, so these are called stereoisomers. One would be R, the other would be S. I don't want to go into how do you know which is R, which is S. Those of you who are interested, I can talk to them separately about it. So here, at least you are able to make a distinction that this is different stereoisomer than the other. Okay? Good enough? Most of the chemical reactions cannot make distinction. Enzymes make a distinction. Enzymes go one step further. Let us say this is CH3, CH2, CH3, CH2, COH, let us say alcohol. These two are hydrogen, CH2, and this is CH3, and back I have COH group. So this is alcohol. Now these two hydrogens, this carbon is not asymmetric. This carbon, where there are 4 different substitutions, this is called an asymmetric carbon. This is not, or a chiral carbon. This is called non-asymmetric carbon. 2 hydrogens which, I don't know, can you make a distinction or not? Probably you cannot at this moment. Enzymes do make a distinction between this hydrogen and this hydrogen. So when you look at alcohol dehydrogenase of, let us say, yeast, so if it removes this hydrogen, particularly in all the cases, it will not touch the other hydrogen, which is almost identical. The molecule looks identical. You have 2 identical groups. Enzymes can even distinguish if there are, let us say, if this is a 3rd hydrogen. They make a distinction between this, this, and this. Always selectively take 1 hydrogen in the reaction. This was illustrated experimentally by a scientist called Venisland. She did that with alcohol dehydrogenase and showed that some of these alcohol dehydrogenases are able to make a distinction or discriminate between what we call as 2 enantiotopic hydrogen. So structurally or stereochemically, these 2 are identical hydrogens. But if you think about it, if some of you are interested, we can still talk about conceptually, people have been able to make a distinction. We can make a distinction between the 2, but I'm not going to talk. But to surprisingly, enzymes do make a distinction between these 2 seemingly identical, which no other chemical reaction can ever think of. So that's the power of specificity. So that's what, that's always amazes me, and I thought I will convey part of that amazement to you. The other is, which I have already, for example, I don't know how many of you were too young, all of you. There was a great tragedy called thalidomide tragedy in 1962. Anyone had heard of this? No. Okay. Not many of you. And the company which made this had to shell out billions of dollars in damages. It was sold as a sleeping pill, and what it caused is it caused teratomas in the newborn babies. So number of pregnant women which took this, the doctors prescribed this as a safe sleeping pill, turned out to be a nightmare for more of them. They had malformed limbs, babies, some of them were dead, and malformed babies worldwide. This was a big controversy. And once it was realized that this is because of one of the enantiomer causes teratoma, whereas the others, as this acts as a relaxing agent. So S enantiomer was tetragenic, but R isomer was an effective sedative. So this realization came, and so people became more conscious, particularly in the pharma field and others, that you have to, if you have to give a medication, and this is, I am talking of only 1 carbon which is asymmetric. Look at, if you look at glucose, there are 4 asymmetric carbons there, and so on and so forth. The bigger the molecule, more the carbon atoms, the number of enantiomers which it will have or increases. So if you have a glucose, how many isomers, stereoisomers are possible for glucose? 16. There, because there are 4 asymmetric carbon atoms, 2 to the power 4, and so on and so forth. So this is the importance of, again, why I am pointing out is this, that in case of one, if you provide the other, it might act on a different pathway, it might have a different effect. For example, some bacteria also make D-amino acid in their cell wall. They add that. So they use D-amino acid. There are some where the L and D act as antagonistics or inhibitors in some enzymatic cases, so on and so forth. So as long as you are aware of the structural importance and the ability of enzyme. Look at this is just a part of metabolism, metabolic chart which I have picked up from a cell. Look at the complexity. The central portion is glycolysis. This one you can see is this TCA cycle. Look at so many of them interacting, the product coming into it, regulating it, and so on and so forth. And all these are catalyzed by enzymes. So hundreds, thousands of enzymes, they have to be fine-tuned, accurately regulated to see what we see. Each cell of us has to undergo this so that we are able to live, speak. I'm speaking to you. At this moment, thousands of reactions are going on each cell of me. So is in the bacteria, in the plant, in the flowers, whatever you see around us, the life. So that's enormous. So with this, I conclude what I thought, if I am able to convey to you, the amazing properties of enzymes, which is strewn all around us in all our cells. We possess them. And that is what makes life possible. Okay, now coming to, this is as far as life as such is concerned, that these are the properties of enzymes. Mankind have exploited the processes based on enzyme. For example, whether it is wine making, cheese making, or anything. If your mother makes curd at home, again an enzyme based process. People have already also knew about the effect of some presence of some enzymes. For example, in the Greek what is called mythology, the book Homer, it talks about the fig milk which can curdle things. So they knew about a protease which is present there. People from ancient civilization knew that calf intestine contains an enzyme renin, which they used to make cheese, variety of cheese in Europe, Mediterranean, and so on and so forth. So people knew about this. Papaya leaves were known, papaya fruits were known to cure ringworms as a meat tenderizer, and so on and so forth, because it contains a papain. So on and so forth. There are n number of such examples. Without knowing, people have been from time immemorial using these systems. But as we know more, we have put them to great use. So what best place to start than to talk about the health field, medical field. Has anybody, one of you has ever fallen sick? Liver disorder? None? A healthy group here. Good. So nobody or any of your family members? No. So good. That's good. So if you are suffering from jaundice, supposed to be, there are tests which are done, confirmatory tests. Doctor would send you to a clinician. Somebody would say, yeah, you will check bilirubin and so on and so forth. So one would like to know whether it is an obstructive jaundice or something wrong with your liver. So what you do? You cut open the liver to see that? No, you depend upon. So what has been done is people depend upon the fluids which I can easily collect, let us say from your saliva, from your tears, from your urine, or if I can draw a blood sample, look at what are the changes in enzymes, some of these enzymes, or let us say if you had a heart attack about last, let us say 24 hours, can I find out? You said I had chest pain. Can I find out that you had a heart attack? Now your ECG looks normal. But can I find out that you had really suffered a heart attack? Enzymes comes to your help. What happens is any organ which is diseased, the cells die there and release the content into your blood. So the blood serum analysis or tells you about some of the medical condition. That's one. Enzymes as such, I don't know, most of you would have heard of the inborn errors of metabolism, right? There are some genetic disorders some people are born with. So they can only be detected if you say that this guy lacks alcohol dehydrogenase, or he is unable to digest lactose. The kid is unable to digest lactose because it does not have lactose dehydrogenase. How do you find that out? The only way you can find that out is analyzing the enzyme. Okay. So inborn errors of metabolism, because and there are n number of such disorders people suffer from. So some cases it could be a brain disorder. Look at the number of enzyme. The way you can detect is that it is lacking is estimate the enzyme. As I said, enzyme in clinical diagnosis in hepatitis, they say SGOT, SGPT, commonly done, or myocardial infarction. LDH is used, acid phosphatase for prostate cancer, and so on and so forth. Also, you would like to distinguish between whether it is problem with the bone or with the prostate. So, if you know the difference between these two so-called isozymes which are produced in different, you can make a intelligent guess. For example, acid phosphatase, prostate enzyme is inhibited by tartrate, erythrocyte enzyme by formaldehyde. So, you know what level has gone up because of which, whether it is because of erythrocytes or is it prostate, or is it alkaline phosphatase placental enzyme, or is it bone enzyme, and so on and so forth. So, studying enzymes, finding out the differences have been a useful tool in identifying number of clinical cases. For example, as I told you about the hours after onset of chest pain, you see the way the enzyme behaves. So, about this is 50 hours, that is after 2 days, you would find that aspartate aminotransferase or lactate dehydrogenase, particularly the heart H4 would go up, and creatine kinase and so on and so forth. So, these enzymes have been made use of. The other I think you are all familiar with, identifying viruses particularly. ELISA has been very commonly used, and ELISA is based on an enzyme. So, an intelligent application of enzymes like alkaline phosphatase or beta glucosidase, which you can easily estimate, have been linked on to an antibody to create a test for various antigens. So, it could be say flu, it could be HIV, you name a virus, not only in animals, in plants also. You can clearly distinguish which is the virus, what is the strain, and so on and so forth. So, devise proper treatment. Enzymes can also be used in therapeutic, as a therapeutics directly. I don't know how many of you have heard of urokinase or streptokinase. If you have a severe clot, people inject these enzymes. They are expensive. They are all, what are they? They are proteases. So, they dissolve the clot. So, enzymes can directly be used. They are not commonly used, the reason being that they evoke immune reactions. But if you have to just give once, you take the risk of using these, and they are used. For example, some of these trypsin and chymotrypsin were used for necrotic tissues, and also asparaginase has been tried as an anti-cancer agent for leukemia, asparaginase injection, because the growing tumor tissues require a lot of asparagine compared to normal. So, if you have an asparaginase, you starve this tumor of requirement of asparagine, and so on and so forth. So, as I said, but large scale use is not possible because they evoke immune response. The number of drugs which you pop in for various disorders, whether it is headache, fever, or your father or mother, if it has a blood pressure, high cholesterol, anything you name, 95% of the medicine which you buy from the market is an inhibitor of a specific enzyme. Why I am telling you is this, that people in the West have been clever. They are continuing their work on so-called studying enzymes and basic biochemistry. All over the world, the other people have moved on with the time thinking, ye to bekar, this is of no good. We have to study molecular biology, clone the genes, talk about genomics, so-called. And in the process, they are making money. We even do not sequence. We ask them to sequence it. We don't know how to read the sequence. They read us, tell us, this is what we means, and we blindly say, okay, agreed. Because we have not developed these technologies, and neither we were involved. We were strong in chemistry and enzymology, but that is past. 1950s or so, we had very good groups. But slowly, because of the changing time, changing funding conditions, We have left them, but they still do that. Most of the money which is being made abroad by all these companies based on enzymes. I'll come to it. If you look at the modern, so-called modern biotechnology itself, is because somebody studied enzymes. Okay, so you name the, so I have few, because I am diabetic, I talk about sitagliptin phosphate. So people have looked at what is the pathway of degradation of insulin, what are the enzymes involved. And they found one, DPP4 enzyme, and sitagliptin blocks this. And this stops breaking of insulin. A number of such steps have been. For example, alpha-glucosidase, which is involved in digestion of food which you take in your stomach. If you take acarbose, which is an inhibitor of this, delays the rise in blood sugar in your. So all these processes, biochemistry has been studied, enzymes have been used. So enzymes as analytical tool also, not only as such, but also enzymes have been used. For example, let us say if, because of their enormous specificity, if I have to know what is the amount of glucose present in my blood, or the concentration of glucose, I have to take the blood, separate the parts, separate glucose from other reagents. Chemically, what we were doing, we have to estimate it. But specificity of enzyme has now given me tool. I don't have to separate the blood from its constituent. I can estimate directly by using an enzyme what is the level of glucose, what is the level of cholesterol in my blood. Use an enzyme because enzymes are specific, and if you can link that specificity with electronics, sensor, whether it is color or electronic signal, a change in pH, change in potential, you can estimate that. So that has been done. So food analysis, similarly in food, how much of ascorbic acid is present? How much of fructose is present in this syrup? Is this Chyawanprash, does it contain vitamin C? How much of it is present? Vitamin E, people say vitamin E, can I know how much it is? Pharmaceutical, environmental, people say here is nitrate in the water. Can I estimate the nitrate? Can I estimate what is the level of arsenate in this soil sample or water sample or whatever you have in mind? Is it contaminated? So all those enzymes have been used as analytical tool. The list is endless. This is the most common one which you have already seen. So just one drop of which is less than maybe 2 microliter is good enough for estimation of blood glucose. So you don't have to draw blood, you don't have to separate anything from it. This strip has an enzyme which is already immobilized onto it, tiny amount which is required for the reaction, and it tells you same thing you can do with cholesterol, the same you can do with ascorbic acid, and so on and so forth. So number of analyte which is required, and people have developed enzymes, cholesterol, glucose, sucrose, galactose, nitrate, you name it, there is a sensor linked on to an enzyme. So enzyme based. Industrial uses, and industry is full of these. And why industries prefers this? To make new molecules not previously possible from chemical synthesis. Look at how you might have seen the structure of, let us say, cholesterol or even a steroid hormone. Look at how many groups are there. So let us say if on position 13 I want to add a CH3 group and it makes a molecule which is novel, I can't do it by chemical synthesis because I have to block all the reactive groups, selectively unprotect, protect, go through number of processes. Enzymes can do it selectively without going anywhere. So that's the power of enzyme. To make improved processes, products, or making a product in cheaper ways, new intermediates to degrade waste, pollutants. Nowadays there is a lot. So enzymes have been used. You know what is a baby food? Companies sell baby food. What it is? It is just milk digested with an enzyme. So there is nothing special about baby food. So the idea is that the digested food is easily digested by the baby. So you are already partially digesting it using trypsin. Baking industry, you name it, fruit juices, cheese, wine, beer. If you want to clarify a product, you use pectinase, number of them, glucose, the Pepsi or Coke which you drink, which is HFCS, high fructose corn syrup, because fructose is sweeter than sucrose and glucose, it's about 2 times sweeter. So what you do is you convert the corn, hydrolyze it into fructose and sucrose, and you find that it is sweeter than sugar. and you get hooked onto it. So all the energy drinks and the colas which you drink, they have high fructose level. So glucose isomerase is used there. Catalase, you talk about it at home, you use detergents. Nowadays all the detergents which you, the advice is that you soak your clothes for half an hour. Why do you have to soak it? Earlier you used to just rub on a cake and wash it. The reason is that those detergents contain enzymes, lipases, proteases, which will dissolve. Most of the stain is because of this, and the dirt sticks to them. So they contain detergents and cleaning powders, leather, baking. You name an industry, you will find, and these are brand names of enzymes which are available by various companies. Alkalase, asparase, Novozyme produces number of enzymes. For flavor, look at the bouquet of flavor which you get when you eat a banana. You know what is a flavor? Or any fruit. These are all aldehydes, and they are special. You can make them artificially released by an enzyme. If you have the precursor, add them, or onto wine, beer, drinks, even fragrances which you use. Enzymes are in demand. Bouquet of flowers. So Novozyme or makes a lot of money selling these enzymes, which are nothing but some glucosidases, esterases, releasing color, and so on and so forth. So you have these variety of enzymes available in the market to make money. So you have taste, dairy, the enzyme, and so on and so forth. You can have a series of, these are the chemicals which give the flavor. These are the enzymes or microorganisms which carry the enzyme. Fruit fragrances, taste enhancer, mustard flavor, garlic flavor, whatever you want. Enzymes are being used. The other one, which is a very important one, which we are not aware, we require amino acids, and particularly if you chemically synthesize, as I told you, you will get a racemic mixture of both L and D. Now, how do you get just L amino acid? Because most of the living organisms just consume L amino acids. So Japanese have come up and they rule the market. They make L amino acid specifically using an amino acid acylase. Do racemization, but constantly convert. It will only act on acetyl L amino acid to release the L amino acid. The other one will go back again. So they use a racemic mixture which is synthesized and use an immobilized amino acylase to release the acyl group to give you L amino acid. And it's an internationally, they hold the patent and this applied. Similarly, different modification of penicillin, which are more potent or have a diverse use, they have a diverse specificity, using enzymes. And this list is endless. For example, how many of you drink diet sugar or diet Coke? NutraSweet, you have heard of it. What is it? It's a dipeptide. It's a peptide between aspartic acid and phenylalanine. It's just a dipeptide. How is it synthesized? It is synthesized by a protease. The protease now adds these two groups, so which is sold as aspartame, which is the constituent of diet Coke, diet Pepsi, or whatever diet drinks you take. So this is synthesized exclusively by a protease, an enzyme. Another, Taxol synthesis. If the one which you get from, if you want more potent ones, what you do is convert these and look at the groups where it can be added, number of places where the reaction can take place. Because of the specificity of this enzyme, you will have addition at only specific groups. So again, power of specificity of enzymes is used. Most of the acrylamide which you get, you use using an enzyme called nitrile hydratase. So you use acrylonitrile and add this, you get acrylamide, which you use in several applications besides research. Industrial application, as I said, sterilization of dairy product, juice clarification. So this list, as I said, is just indicative. A list is endless. Now coming to environment cleanup, pesticide, petroleum residue, enzymes are being used to clean up in situ, ex situ, all those water treatment. If you have nitrate, you can use nitrate reductase and so on and so forth. So you have these enzymes have come in as the advantage is they are safe, they are biodegradable, so there is no toxic effect. So enzymes are preferred. The other, as I said, industry prefers them because you don't need higher temperature, higher pressure, so the energy requirements are less. Now, an enzyme-based technology which has almost revolutionized all the branches of, without which other branches, as we see, this is one technology which I know of besides the silicon chip, which has spread all over with our electronics. This is one enzyme-based technology which we overlook, which has impacted the genetics, environment science, forensic. Know what is this technology? An enzyme-based technology which has given birth to new fields and new fields, newer fields. You are all aware. Unfortunately, like I said, it is so common that you think, oh, ye to pata hi tha. Yes, PCR, yeah, a polymerase-based system. So look at in veterinary sciences, you name it, including from archaeology. You recently would have seen a news that a girl in Denmark, 40,000-year-old, chewing gum, the gum which she had chewed and spitted out, they have isolated DNA and made a picture of it, how she looked, what she ate, what was her last meal, and so on and so forth. Amazing. Don't you think so? Again, this is possible because you have DNA technology. Our number of, earlier we used to say for a child that maternity is certainty, paternity is a matter of opinion. But now with DNA technology at your hand, you are now able to make paternity also a certainty. So there are a number of court cases which have been settled using enzyme-based technologies, whether it is restriction enzymes or markers. So all this is possible because of, I don't know how many of you heard of Lalji Singh. He has he has been as a witness, a number of court cases because of his evidence which he has produced about murderers, about victims, about even the dead body which was tuned over, which part is which, whose body part is this, and so on and so forth. Famous Rajiv Gandhi murder, he was able to identify the tissues, and so on and so forth, including the so-called new biotechnology which exists, for which you have all come. It would not have been possible if there were no enzymes, no restriction enzymes, no DNA polymerase, no alkaline phosphatase. You would not have been here. Okay, so enzymes used in the list is endless. If you are doing a transformation, you are looking at blue and white colonies. What is that enzyme? Which is a beta-glucosidase. Or in plants, you are using glucuronidase. So again, enzyme-based selectable markers, NPT or whatever HPT you call it, selectable markers, screenable markers. You think of anyone, even the earliest proof of that DNA is genetic material. Have you heard of every McLeod? Yeah. What was their evidence to suggest that DNA probably is the genetic material? They used enzymes, proteases, lipases. So look at this. Even to prove at that time, enzymes were used as an analytical tool to confirm what they are saying is right. So it's as old as 1940s, the use of enzymes for proving a point. Some of you would have heard of protoplasts. And how do you create protoplast? You have to degrade the cell wall. So how do you degrade cell wall? Again, enzymes. So one of the technologies which you talk of as somatic hybridization or protoplast fusion has come because of use of enzymes. Again, all these enzymes you import. Japanese have mastered the art of enzymes, use of enzymes and preparation of enzymes. This is again, I'm repeating them. So golden rice, I think you have all heard of it, that this is a technology which you can produce in rice, endosperm, beta carotene. Now this has become possible because people looked at what are the steps in biochemistry that are missing in this pathway in rice, and can I now introduce these into rice with specific promoters so that they are able to express in high amount beta carotene. And so you have phytene synthase, lycopene cyclase, phytene desaturase from different, earlier it was from narcissus, now it is from maize. So from a bacteria, and people have now worked on, so a rice glutamine promoter from maize and assembled and produced, which is a reality. Probably it will be released in Philippines this year, and Bangladesh is contemplating releasing golden rice. The disadvantage is that without this, number of children who depend solely on rice go blind every year. So that probably would be stopped. Here, I'm not going into, if you look into literature, you will find people have worked on, can I increase the level of vitamin E in grains? You are here for quality. So people have increased ascorbic acid, vitamin A, folate, which is a very major problem with lactating and pregnant women. Folate has been increased in maize, and so on and so forth. So people are working on it, again based on an enzyme which converts the precursor into the product. The so-called genome editing, which you have heard of so far, all of them, earlier also, when we were using zinc finger nuclease, or whether we were using TALEN, and now it is CRISPR-Cas. These things keep on changing, but one thing which is common is an enzyme is required. Now people are working on improving Cas9. While we would start the ground zero, the old Cas9 which is available, they would have already gone into an improved one, and so on and so forth. So there is a rate which is a race which is on, and we are into the rat race. But still, all these technologies, future is with enzymes. You have heard of blue rose, okay? Scientists have been able to create, again, the importance of biochemistry and enzymes, understanding what are the steps required, what all the pigments which would be produced by these enzymes. They are able to create a variety of colors in these transgenics. So blue rose is a reality which is being sold by a Japanese and Australian firm. This is another one which I find quite intriguing. Now the enzyme Luciferase you are already familiar with in Firefly. Now there are people who are working on, can we make plant express this? I don't know whether you can see here, there is a face, probably not, of a person. But anyway, so these are experimental plants where a luciferase has been made to express. So 20 years from now, I'd not be surprised if you have pavements lined with plants which would glow in the dark, or begin with, you will have, instead of a night lamp, a plant, the moment there is light is switched off, it will glow. And so that's the way we are going. So it's free from, it uses solar energy, produces light for you when it's needed. So we have been exploiting plant since time immemorial. Now we have learned to exploit them in a more refined, more defined way to suit our own need. Here is one example. So look at where the science is going with study of enzymes again. Here is the, I think I would say probably this is the last example. Here is an example. This I would not have said would have been possible say 10 years before if you had asked. Now this is a plant, May apple, which is used for the podopholotoxin which is produced by this is used for cancer treatment. It's highly sought after and it's highly endangered because it's grows in some areas of Himalaya and it's slowly becoming distinct. So people have understood what are the steps which are required for synthesis of this, and can we introduce these steps into tobacco. And this paper appeared in 2015. So this 6 steps, 6 enzymes have been introduced into tobacco. Now the tobacco produces this podopholotoxin, and So in one step, what you have been able to do is, and it produces a different version which is more potent, so that's added advantage to them, but you have also been able to save the plant. So you are able to add towards conservation of plant as well as produce something which is of value. So again, how that has been possible, because people will say we had the gene. It is not the gene. People understand the biochemistry, looked at the enzymes, and then cloned the gene. Genes by themselves would not do anything, this is the enzymes which are doing it. So we have our mindset towards genes. We don't look at the products, which has been unfortunate for us. Look at, people talk about carbon dioxide. Level is increasing. Why is it increasing? Who fixes this carbon dioxide? Again, one of the enzymes which is present all over the world in large amount, Rubisco. So again, importance of enzymes in our life. This is the example which I said. So you have a 6,000-year-old chewing gum which they found, and they are able to create an image of Lola by that, because you are able to now read the sequence, read what would be the face like, whether her eye color would be blue or brown, what would be her hair color, and so on and so forth. But all this, I am not saying that is because, but it originated because you are able to now at least clone the piece of DNA which was present, which was already existing in most of these 80,000 old mummies or Neanderthal DNA which we find. We are able to identify from that, and that has been possible because of enzymes. I don't know how many of you remember 2018, nobody talked about it. This lady, Frances Arnold, she got Nobel Prize. And for what? For the directed evolution of enzymes. The idea is that if in nature enzymes have evolved because of certain reasons, so they are dependent on a substrate, they want to convert it, make energy out of it. So people now think of, can I now direct this evolution, make it faster? Can I get an enzyme, let us say if in yeast strain, which will produce butanol, which I will use, otherwise it only produces ethanol. So she has not only this, she has done number of other work and developed the technology for it. Can I now, there is another group which got together with her, because, which is again used for this purpose, which is called phage display. So you have several antigens displayed and you select those. So she had made use of, and she got Nobel Prize in 2018. But probably most of us are not aware of it, that this is for directed evolution of enzyme. So the current scenario, this is the last slide, is that you have, if you have a process which you have in mind for industrial use or any other application in plants, is the reactants and products, if you know the enzyme. If you know, very good, you go to whatever it is, this is the path. So is it readily available? If not, you clone it. It is adequate activity. If any of the answer is no, that the enzyme is not known, adequate activity is not present, it is not stable, process is not developed, you can screen and test for the activity. There is another path for this, that if you know the substrate and you have an antibody for it, so people have, so at least specificity is taken care of. Now people have tried to convert these antibodies into enzymes, so which are called as enzymes. So that's another path which follow. So if not, you can mutate the DNA and have variation. So create what otherwise nature would have done. You enhance the process of able to create mutations, get whether it's the enzyme is stable, you want to use it at 90 degrees, but the enzyme is very unstable at 100 degrees. Can I make it stable? Yes. So people are working on it. If the industry requires that I have to freeze it, can enzyme withstand freezing? You can change it. Do I have enough activity? You name a question and the answer is you can do it. Now there is a famous phrase which is going nowadays around. I have modified it, though that phrase is controversial, but I have modified it to say that enzyme hai to mumkin hai. For those Tamilian students, You know this phrase. No? You have not heard of it. Okay. Sensitive phrase, I'll talk to you about this. But the question is, if enzymes are present, ye luma, ye lada. Ye lum. Okay? So enzymes can do it. If you have a process in mind, the enzymes can make that happen. And with that, if you have any questions, I'd be happy to answer them.
So if you have any queries, please come in front and then ask.
You need not come here, you can holler from there also. It doesn't make a difference. Yes, you had some question, no? Okay, good enough. No?
So it was something like I was sitting and I was thinking that it should go on. There should not be the last slide. Thank you. It was so interesting. And it was so interesting. And I think I should add, because I have seen different sets of sir. He is excellent in literature, excellent in science, excellent in presentation, whatever area you select. And sir will love to talk, love to present. And he will give an impression that it's difficult to then, because he will try to touch each and every dimension of that particular topic. Let's see today's topic, enzyme. Even I have never heard such an exhaustive talk on enzymes. And I think you all will agree with this. So with this brief note, now I request my coordinator, Dr. Goswami, to please felicitate sir with a memento.
Thank you very much. Thank you very much.
Sir, on the behalf of this batch, so I would like to thank you for giving such an interesting and then exhaustive talk. I think definitely we'll have one more schedule with sir, if time permits. Thank you. Thanks a lot.
Thank you. Thank you.
One more thing.
Yeah. Acha. Thank you. वो रखवा दीजिए, उसको भिजवा दीजिए, ये डब्बा भिजवा दीजिएगा उसके साथ ही.